Hello, everyone. Welcome to my Nanostring Spatial CosMx data analysis in Seurat. For today's demonstration, we are going to use a dataset generated by Nanostring from human non-small cell lung carcinoma. You can download the dataset from Nanostring website. Here is the data summary. You can see they used the 960 gene target and analyzed the 8 tissue sections. You can see other information about the total tissue area, the number of field of view, total cells, and also down here you can also see the donors information. So the full dataset contains 8 different samples from 5 tissues. The tissue are from 3 female and 2 male, and all the donors are age over 60, and also here are the tumor grade. So if you click the download data link, then you can download all the data files from this link. For the demonstration, we are going to use the num file repeat 1. Then you can click here the link to download the data. So we are going to analyze the data in Seurat. For analyzing the data in Seurat package, you also need to download the coordinators for the cell boundaries. You can use this link to download the coordinators. As I said, for today's demonstration, we are going to analyze the data in Seurat. You can use the other package to analyze the dataset. For example, the Nanostring company developed a method here. You can use this method to analyze CosMx spatial dataset. So I downloaded the data. You can see in here in this folder named the num file repeat 1. So you can see 4 folders: cell composite, cell labels, cell overlay, and the compartment labels. If you open each folder, you can see each folder contains 32 images. You can see in the cell composition folder, there are 32 images for the staining. And also we can open the cell overlay folder. Let's open one image. You can see in those images, they labeled the cell boundaries. So when you download the data from Nanostring website, the data also includes the expression metafile, field of view positions, metadata, and the TX file. But this one, the polygon file, is the coordinators file that we downloaded from the link on Seurat website. So we have the data here. Now we can load the data into R to perform the analysis. First, we need to load the packages Seurat and then tidyverse. So Seurat package has a function load the Nanostring function to load the data generated by Nanostring. So in this function, we can specify the files directory. You can see here is the file name num5 repeat 1. I just show you all the data in that file. And we can name the field of view as num5 repeat 1. Then assign as the Nanostring. So now we can use this function to load the data for the num5 repeat 1 dataset. So let's run. Okay, you can see we loaded the Nanostring spatial dataset in the data windows, and we named it the nano.object. So because this is a Seurat object, so first we can have a look at the row names. We know the row names are gene names. Let's run. So you can see the gene names from the row names. On the Nanostring website, it says 960 genes. But here you can see the row names have 979 genes. If you look at the Seurat online tutorial, for this dataset analysis, they used the Asmus reference dataset from healthy human lung to annotate the cell clusters. Because the data was generated from non-tumor, so actually the tumor has different cell composition from healthy human lung. So we are not going to use the Asmus reference dataset to annotate the cell clusters. We are going to perform unsupervised data analysis to identify the cell clusters in this dataset. If you want, you can use the standard Seurat workflow to analyze the cell clusters. For the standard workflow, you can use normalized data, scaled data, and find variable features. Here, we are going to use the SC transform method to perform the data normalization, scale the data, and find the variable features. So this is the improved method for data normalization. So let's run SC transform for the Seurat object first. Following SC transform analysis, we can run PCA. So next we can run UMAP. So I'm using MPC as 50 as I found that 50 can generate similar Seurat clusters as the online analysis from the Nanostring website. So after UMAP, we can find the neighbors. Now we can find the cell clusters. I'm using the resolution as 0.3. If you want to practice, you can change the resolution to see how many cell clusters you can identify from your analysis. So you can see at a resolution 0.3, we identified 13 cell clusters from this dataset. So now we can use the dim plot to look at the cell clusters. Let's run dim plot. And you can see we have 13 cell clusters from the UMAP. The reason that we want to perform spatial data analysis is that we can see the cell clusters and the cell types on the image. So now we can use the image dim plot function to look at the 13 cell clusters on the tissue. Let's run. So you can see we generated the image to show the cell clusters on the tissue sections. For example, you can see cluster 0, they are in blue color. Later from our analysis, you will see cluster 0 are the tumor cells. So we have 13 cell clusters here. As I mentioned, we are not going to use the Asmus reference dataset to label the cell clusters. If you look at the online tutorial from the Seurat website, they have the cell clusters named, but because the tissue is from non-tumor, so actually I think lots of cell types are not correctly labeled. Using the healthy lung reference dataset. For example, if you use the reference dataset, it labels the tumor cells as basal cells. So we are going to use the traditional method from the Seurat package to label the cell clusters here. So if you are not sure, we can use the find all markers function to find the markers for each cell cluster. So we can run find all marks function. Here I set the minimum percentage expression as 25% and the log fold changes threshold as 0.25. So let's run this function to find the markers for each cell cluster. So you can see R is calculating the mark genes for each cell cluster. First is cluster 0. The next is cluster 1 until cluster 12. Okay, you can see we found the mark genes for each cell cluster, and we generated a data frame named as all markers. So we can click this data set to look at the mark genes for each cell cluster. You can see here are the mark genes for cluster 0. It has like KRT19, KRT17. So according to the online tutorial from the Nanostring website and also the Seurat package website, cells in cluster 0 are tumor cells. So you can find the markers for cluster 0, then cluster 1. Then we can select the marker genes from each cluster to do feature plot or variant plot to see the marker gene expression in different cell cluster. For example, we can use the feature plot for KRT19 and KRT17. It labels cluster 0. So we know cells in cluster 0 are tumor cells. Let's run the feature plot. And we can zoom in. You can see cluster 0 express high level KRT19 and KRT17. So we name cluster 0 as tumor cells. For example, we can also use the variant plot to see the expression of EPCAM and LAMP3. So EPCAM is a cell marker for epithelial cells, and LAMP3 is a marker gene for AT2 cells. So let's run the variant plot for EPCAM and LAMP3. So let's zoom in again. So you can see EPCAM and LAMP3 label cluster 10, and also EPCAM label cluster 0. Because this is a non-epithelial tumor, So tumor cells were derived from epithelial cells, so all the tumor cells can be labeled as epicam. So now from the wiring lymph node analysis, we know cluster 10 is epithelial cells. Then we can use like ACTA2, NAM, they are fibroblast and smooth muscle cell markers to show cluster 2 are fibroblast and smooth muscle cell. So let's zoom in again. You can see ACTA2 labels cells in the cluster 2 and also NAM is a fibroblast marker label cluster 2 as well. So we know cells from cluster 2 are fibroblast and smooth muscle cells. So I list the other marker genes here. I'm not going to run through all the feature plot, but for example here, PDGFB and TAGLN labels LV smooth muscle cells. LV smooth muscle cells is in the cluster 12. Then we can use PCAM1 and RGS5 to label endothelial cells and the parasite. They are in cluster 5. So the rest are immune cells. If we use CD14, C1qa as a mark gene, we can identify cluster 1 as macrophages, and mark genes for monocyte are FCGR3A, CD68 to label cluster 9 as monocyte. Because the dataset only has 979 genes, so it's more difficult to use classic mark genes to identify different cell clusters. And we can use 108 and A9 to identify cluster 4 as neutrophils. We can run this feature plot. And zoom in, you can see it's 108 and A9 labels cluster 4. So use all the mark genes down here. We can label cluster 4 as neutrophils, cluster 3 as T cell and NK cells, cluster 6 as B cells, and cluster 7 and 8 as plasma B cells, and cluster 11 are the cells for master cells. They express TPSAB1 and TPSB2. We can run this one again as an example. So you can see cluster 11 express high level TPSAB1 and TPSB2. So using this method, we can label the cell cluster now. Cluster 0 as tumor cells, 1 as macrophages, 2 as fibroblasts and smooth muscle cells, 3 is T cells and NK cells, 4 as neutrophils, 5 contains endothelial cells and parasite, 6, 7, 8, they are B and plasma B cells, cluster 9 as monocyte, 10 are epithelial cells and 11 as master cells, cluster 12 are the LV smooth muscle cells. So we can name the cell clusters. Use the rename ident function to name all the cell clusters. Let's run. So we named the cell clusters now. Let's run the dim plot again. And we can zoom in. You can see now, here is the tumor cells, here are the master cells, here are the B cells, then endothelial cells and the parasite fibroblasts and the smooth muscle cells. Here is epithelial cells. Down here are the monocyte, macrophages, neutrophils, and T cells and NK cells. So let's have a look at the cell clusters from the online analysis. You can see the UMAP from our analysis are different from the Seurat online tutorial because they use a reference dataset from healthy lung tissue to label all the cell clusters. If you look at the cell clusters from the Nanostring website, they analyze the cell clusters using their own method. So you can see our cell clusters are very similar as the Nanostring data analysis. You can see here they labeled, here are the tumor cells, then mast cells, here are the B cells, then T cells, epithelial cells, fibroblast and endothelial cells. Here are the neutrophils, here are the macrophages and the dendritic cells. So we didn't separate the dendritic cells from the macrophages. The cell cluster organization from our analysis is very similar to the Nanostring data analysis. So now we labeled all the cell clusters, then we can use the image dim plot function again to look at the cell clusters on the tissue sections. Let's run. And we can zoom in. So you can see we labeled the tumor cells in blue color, macrophages in red, then fibroblast smooth muscle cells as green, and LV smooth muscle cells as light blue, then the rest are immune cells. So we cluster the cell clusters use unsupervised analysis, and also annotate the cell clusters using the find all markers function, feature plot function, and the variant plot function. So spatial data analysis has great advantage to see gene expression on the tissue section and also to show the cell cluster localization on the tissue sections. So now we can use different visualization method to look at gene expression and the cell cluster localization. For example, we know the tumor cells express KRT17, so we can use the image feature plot function to look at the KRT17 expression on the tissue. Let's run the image feature plot for gene KRT17. We can run. So you can see on the tissue sections, KRT17 is highly expressed in those cells we know they are tumor cells. And also you can use the image dim plot function with the argument molecules as KRT17 to see KRT17 expression on the tissue sections. We can run this function again. So you can see now KRT17 is expressed in tumor cells in red. So here we only show the one gene expression. You can use the molecules argument to see different gene expression. For example, we want to show KRT17 for tumor cells, C1KLA for macrophages, IL-7 receptor for T cells, and ACTA2 for smooth muscle cells. We can run the image dim plot function for 4 molecules. Let's run. So you can see we generated the image on the tissue section to show KRT17, C1KOA, IL7R, and ACTA2 expression on the tissue sections. Each gene labels a different population of cells. So here we use the 4 genes to show their expression in different cell types, and also you can use the image dim plot function to see co-expression. For example, we know KRT17 and KRT19 are expressed in the tumor cells. If we run the molecules as KRT17 and 19, we will see the co-expression of both genes in tumor cells. Let's run. So you can see we generated the co-expression image for KRT19 and KRT17 in tumor cells. KRT19 is in red color and 17 is in blue color. Both genes are highly expressed in the tumor cells. So here I use some gene as example to show you can look at gene expression on the tissue sections. If you like, you can also to see the cell types on the images. For example, we can use the image dim plot and set the items as tumor and B cells. Then we can see where are the tumor cells and the B cells. Let's run. So you can see on the tissue sections, the tumor cells are in blue, B cells are in red. So you can see on the tissue sections where different cell types localize. So we run the tumor cells and the B cells. We can also to see the localization on the sections for tumor cells, macrophages, and monocyte. Let's run. You can see now the tumor cells are still in blue, macrophages are in red, and the monocytes are in green. You can see most monocytes are localized in this region. We can run more example to see the tumor cells and the neutrophils. Let's run. So again, you can see tumor cells in blue, neutrophils are in red. Most neutrophils are in this area as well, like the monocyte. So I showed you how to look at gene expression on the tissue sections, and also to look at the localization of different cell types on the tissue section. So if you want, you can also zoom in into an interested area. To zoom in, we can run the image dim plot function again. Here we set the axis as true. Then we can see the labels for the axis. Let's run this function first. So now you can see from the new image, we have the numbers showing on the x and the y axis. We can use the position of x and y axis to zoom in into a small tissue area. For example, if we want to zoom in this tumor area, we can set the x region from 155,000 to 165,000. Then set the y-axis as 8,000 to 20,000. Then we can crop the image to zoom in that region. Let's crop the image. Then we can set this area as zoom 1 area, and also set the default boundary for zoom 1 area now. So after that, we can use image dim plot function again to see the area that we are interested in. So now the field of view will be the zoom 1 region. Let's run the image dim plot function again. And we can zoom in, make the image even bigger. So now you can see the cell types on the tissue area we zoomed in. You can see tumor cells, macrophages, and also you can see the boundaries for each cell on the tissue sections. So we can see the cell types in this area we zoomed in, and also you can look at the molecules expression in the zoom 1 area. For example, now we can set a molecules argument as KRT17, C1QA, and PCAM1 as an example to see the gene expression in the tissue area that we zoomed in. So we can run the image dim plot function. So you can see now we generated an image in the area we zoomed in. We still label all the cell types here, and also we can see KRT17 expression in red, C1QA, they label macrophages in blue, and the PCAM1 is a mark gene for endothelial cells. Okay, I use some gene names and also cell types to do the demonstration. You can try to look at the gene expression for other genes on the tissue and also the localization of other cell types on the tissue. So I'm going to stop here for the demonstration. I hope this video could help your data analysis if you have a dataset generated by Nanostring. So please subscribe my channel if you haven't to do so, and I hope to see you in my other video tutorials. Thank you.